Hello, everyone. Thank you for coming to today's workshop. I will present my work, CryoREAD. CryoREAD is a method for de novo structural modeling on nucleic acids in cryo-EM maps using deep learning. First, I will briefly introduce the background of structural modeling. When we talk about structural modeling, that typically means we have 3D microscopic map from different technologies, and we hope to use some structural modeling tools to automatically determine the atom locations of different nucleotides or residues of the structures to fully build the full atom structures. There are typically 3 ways to determine macromolecular structures. The first is X-ray crystallography. That's to say, we first crystallize the structures, and when X-ray beam cross those structures, the crystallized atoms will have different directions to different. The X-ray will diffract to different directions. Will nucleic magnetic resonance in the structures, the different atoms will have different frequency under the nuclear magnetic field, and that can help us to indicate the atom types as well as the atom locations. For cryo-EM microscope, we will first make cryo the structure, and then the electron beam will cross the structure to 2D maps, and we use a lot of those 2D slices to construct 3D maps to help us determine the final structures. If we compare those 3 methods of deposited structures of every year, if we look at the right figures, the x-axis is the year. Each year, the y-axis is solved structures of different methods indicated by different colors. As you can see, cryo-EM structure grows exponentially compared to other tools. In 2010, it's very few structures are determined by cryo-EM. Well, in 2020, almost half of the structures are determined by cryo-EM. That's to say, cryo-EM becomes more and more popular. Compared to other technologies, there are at least 2 advantages of cryo-EM. First, we don't need to crystallize the structure. That is a very complicated process. Also, we don't need to, cryo-EM can help us to determine very large macromolecules. I will briefly introduce the cryo-EM structure determination pipeline. So first, we collect samples by experiments, and we put those samples in the grid. Then we freeze those examples and send it into electron microscope. And by the electron beams across those structures, we get a lot of 2D projections in different angles. And we cluster and pick some particles by particle alignment and averaging. Then we reconstruct the 3D cryo-EM maps. And what we aim to do is to develop tools that help us to automatically determine 3D structures from the 3D map. So in short, our idea to do here is that structural modeling for cryo-EM maps means that taking the 3D cryo-EM map as input, we hope our tools can automatically determine the structures, which means we can automatically to determine the 3D coordinates of many atoms and different atom types on different positions. But there are a big challenge for us to determine structures for cryo-EM maps. The challenge is the resolution constraints. When we get those 2D projections to 3D maps through the peak particles process and particle alignment averaging process, that will encounter some difficulties, since some regions may have limited 2D projections and some 2D particle alignment is not so successful. That causes different resolutions of the 3D EM maps. Briefly, we can divide the cryo-EM map resolution in 3 different levels. The first level is near atomic resolutions, which is higher than 5 angstrom resolutions. That we can use deep learning or other technologies to determine the atom types as well as residual nucleic types based to help us to do full atomic structure modeling. And for intermediate resolution is from 5 to 10 angstrom structure resolution. For that resolution, we can't expect we can determine atom details so well. Well, we can still determine some structural information, whether this region is protein or whether this region is DNA, RNA. When the resolution continues gets worse, when it's below or even lower than 10 angstroms, that is hard for us to even determine the rough structure and secondary structures. We can only expect to get the overall shapes. Therefore, today our focus will focus to 0 to 5 angstroms for DNA-RNA structure modeling, where we can expect that our deep learning technology can help us to detect some informations, and those information can help us to use for DNA-RNA full atomic structure modeling. So let's move on CryoREAD. CryoREAD is a tool that we published in Nature Methods this year, which use deep learning to help us to build full atomic models of DNA, RNA in cryo-EM maps. So in short, the basic idea is that we take the 3D cryo-EM map as input, and we hope CryoREAD can automatically construct DNA, RNA structures. For structural modeling approaches in cryo-EM, we can roughly divide that into two categories. One is manual modeling, another is based on computational tools. For manual modeling, people typically will use some interactive tools such as CoDE to visualize the density details and manually put those nucleotides or residues into corresponding regions. But that is very time-consuming and can risk a lot of human error since it's a very labor-intensive work. And another direction is to use computational tools to do structural modeling. There are very few works that develop tools for structural modeling on DNA and RNA. Two tools is HARO-SPEX and Phoenix. Well, HARO-SPEX can only achieve protein secondary structure and DNA and RNA information detection. And Phoenix is based on fragment assignment-based structural modeling tools. It's originally designed for proteins, so its DNA RNA structure modeling is quite bad. If we compare Phoenix model structure as well as the native structure in the right, you can see it can't even locate the DNA RNA regions very well, not to mention to model it accurately. In short, up to now, there is no good DNA RNA computational tools. To address this need, we propose CryoREAD, which takes cryo-EM map as well as optional sequence to automatically build the DNA RNA structures. Compared to previous tools, it's fully automated and it can build real model structures. And based on deep learning, it's quite fast and smooth. First, I will briefly introduce the background of DNA RNA structures. The basic component of DNA RNA structure is nucleotide. In nucleotide, there are three main components, which is phosphate, sugar, and base. If we take a close look at DNA RNA nucleotides, we can see they exist very minor differences. They exist very minor oxygen differences in sugar regions, and they share three same base types, where timing and uracil is different across DNA and RNA. Well, they only exist very minor differences of the carbon in the left top bottom region. So based on that, we can roughly think that we can treat DNA RNA as the same categories and do the DNA RNA structure modeling as the same direction. The basic component of nucleotide will connect together to form the phosphate sugar backbone by the dehydration process, which the oxygen as well as the oxygen in the sugar will concatenate, will have the dehydration process, and two consecutive nucleotides can connect together. And after they connected, different nucleotides will share different base types that result different nucleotides in the sugar phosphate backbone. And different base types can form the base pairing, such as A and T, C and G. And based on the base pairing, it can form a relatively stable 3D structures of DNA and RNA. Inspired by the basic component of DNA, RNA, and how it conforms into the 3D form. We developed our own framework of CryoREAD, which also starts from very small nucleotide and continues to trace the backbone to build the full structures. So first, we will use deep learning tools to segment the 3D cryo-EM maps into different grade-wise detections, segmentations, with sugar phosphate-based detections. And based on the sugar phosphate detections, we will trace the sugar phosphate backbone. And after we have the backbone, we use our deep learning tools to detect the base types to assign different base types into the different nucleotide. After we get the sequence assignment, we further build the full atomic structures from that. So in the step 1, we first use a cascaded unit to take the 3D cryo-EM map as input to output the segmentation of the EM maps. So to achieve that, we use the box to scan across the maps. And for each scanning, we will output a corresponding probabilities of different classes, sugar phosphate base type classes in the box, and we combine them together to have the full phosphate, sugar, and base detections. The network architecture is shown here. It's a cascaded unit. So the first unit will in charge of big class detection. That's to say, it's focused on how do we detect phosphate, sugar, and base, as well as proteins. While the second unit will take the information processed by the first unit as input, as well as the density as input, to further predict the base types in the base regions. So in short, after the processing of the cascaded unit, it will predict our segmentation of the sugar, phosphate, and base, as well as the base type detections in the base regions. After that is finished, we further train the refine unit, which takes the sugar, phosphate, base, protein, as well as base type detection probabilities as input, and to further refine the probabilities to give the final outputs. The basic idea is that we hope to take the neighboring probability predictions as input, to take the neighboring information to help us to refine the predictions. If we compare the detection of stage 1 and stage 2, you can see the accuracy increased generally for all the classes from stage 1 to stage 2. In stage 1, stage 2, we can achieve more than 80% for big classes detection and more than 50% of small base type class detections. When we have those accurate detections, we will focus on how to trace the backbones. With the sugar detections, we first use mean shift to clustering to get those representative nodes. And with those representative sugar nodes, we connect them to sugar graphs by geometry constraints as well as the initial sugar detection results. With the sugar graph, we hope that we can trace the sugar backbones from the graph. That's very similar to trace the path from a complicated graph in computer science. Well, for this example, it seems to be a quite easy task. But if you look at more common examples in our dataset, those very big graphs, it's very hard to trace the backbone accurately. Therefore, we propose to reformulate it as a vehicle routing problems to solve the problem. Vehicle routing problem is an upgraded version of traveling salesman problem. In traveling salesman problem, that's to say I have a big graph, have n locations, and we ask a traveling salesman to visit all the cities, and we want to minimize the cost to visit all the cities. Similarly, vehicle routing problems, we have n vehicles to visit all the locations, and we still want to minimize the cost. But compared to before, that allows us to drop some points or locations that we think is unnecessary. If we consider the sugar nodes as locations in vehicle routing problems, and if we have NP chains, we consider we want to have NP vehicles to visit those locations. And when we want to drop wrong nodes, that's very similar to apply a drop penalty for location here. So that's to say we successfully reformulate the problem as a vehicle routing problems to trace the backbones, like we visit a path of n locations. After we trace the sugar backbone, we want to check what should be the base type of different positions of the sugar nodes. So with the previous unit, we can get the base type detection in the base regions. Then we take the sugar backbone as well as the sugar detections to have the initial base type assignment by simply take the closest base type detection to the sugar nodes. But that has some problems here. First is that two sugar nodes are too close. Well, in real cases, two consecutive nucleotides should satisfy some geometry constraints. That to say, they typically will be 5 to 7 angstroms of two consecutive sugars. And that information is not enforced here. Also, we sometimes can hopefully get the sequence information, but we did not use that information in our training. To address that, we want to do something to help us to get better sequence assignment. So we take the sequence and the initial sequence assignment as input. We use a sliding window to move across the initial sequence of assignment. So that's to say for the traced path, I get a lot of scanned fragments. Then by considering the sequence and the geometry constraints, we can have dynamic programming. That's to say for one fragment, I can have a lot of possible fragment assignments with different scores. After that process is finished, that's to say I have a lot of overlap fragments, and each fragment I have a lot of candidate sequence assignment. But there is some conflicts of those assignments. First, for one fragment, in the end, we only expect that we have one final sequence assignment. And another thing is that for overlapped LDP as the path regions, we hope the assigned sequence are consecutive. And for consecutive sequences, it should assign to the same consecutive regions instead of distributed in two regions in the trace main chain. So to address that, for different sequence assignment, we have a score by the dynamic programming, and we want to sum up to get the highest reward. And then the problem can be reformulated as a constraint programming task. That's to say, for those conflict assignments, we only ask them to at most pick one and should pick one for that. And by optimizing this problem, we can have the final assignment. That's to say, with the initial assignment taken from the base types, I use dynamic programming to get a lot of assignment for each fragment regions. And with that each fragment regions, I further do the constraint optimization to optimize the overall reward to get the final assignment. After we have the final sequence assignment, we check the neighboring phosphate and base node to have the sugar phosphate base triangles. And if we check the standard nucleotide sugar phosphate base triangles, we can use those geometry information to place different standard atomic structures along the sugar phosphate base backbones so that we can have the atomic structures. Then after the framework of CryoREAD build, we benchmarked on on one data set with many cryo-EM maps. We measure it across two measurements. First is the backbone recall. For backbone recall, we just check how many nucleotides in the ground truth have nearby predicted nucleotides. And for that, the backbone recall is more than 80% of CryoREAD, while for Phoenix, it's only around 50%. And another measurement is sequence recall. That's to say for the ground truth nucleotide, I will check if neighboring predictions have the nucleotide by CryoREAD, and the CryoREAD can assign correct base type for that nucleotide. For that, CryoREAD achieved around 50% sequence recall, while Phoenix only achieved around 20% sequence recall. Here I show some examples of CryoREAD. The first is DNA examples. When we take the EM map as input, CryoREAD can build the structures, can achieve a very accurate sugar, phosphate, and base detection, as you can see in the middle. Based on that, we can build the full structures in the right. It achieved the backbone recall of 86.2, which is very identical to the ground truth structures. The second example is the RNA example, where the model structures by CryoREAD is shown in the middle, while the native structures, which is ground truth, is shown in the right. You can see the overall conformation and base detection assignment is very accurate into the correct positions and correct orientations. It has achieved a backbone recall of 90.9%. In short, in these tutorials, I briefly introduce our CryoREAD. CryoREAD can simply take EM map as well as optional sequence information to build DNA RNA structure accurately. It's fully automatic and it can deal with multi-chain structures as well as protein DNA RNA complex. To simply use our tool, you can scan the QR code to use our server online, which is free and process jobs quite fast. You only need to upload the EM map and sequence, then you can get the DNA RNA structures modeled online. You can also try Google Colab with step-by-step instructions. In the end, I want to thank all my lab members, especially Professor Daisu Kahara and Genki Terash. Without their kind supervision and help, I can't do such great help. I also want to thank my fellowship supported by MOSE, as well as the funding from NSF and NIH. Thank you, that's all.